need to talk about, because this is MSK block, that little thing is not here. Yes? Okay, so I need to talk about skeletal muscle. Now, you had a brief introduction during your foundation block on muscle, and I think it was mostly cardiac muscle, which is kind of similar to skeletal muscle. And then you did smooth muscle, am I right? Okay, and we're going to build on that a little bit. But this time specifically with skeletal muscle, because cardiac muscle you kind of studied in detail during cardiovascular block. You did a little bit of smooth muscle thing during GI block. You kind of got a sense of how those things worked. Now it is MSK block and we need to talk about skeletal muscle. And as the name suggests, it's muscle that's attached to bones or to the skeleton. And since all muscles have this ability to become shorter, they have the ability to become shorter. If the muscle is attached to a bone, that means the bone is going to move, or at least the joint is going to move. All right? And therefore you're going to get locomotion, or you're going to get movement, and so on and so forth. So we need to look at this skeletal muscle thing. And today's lecture is reasonably simple. It's just a recall of stuff you may have done before. Again, as the name suggests, it's muscle that's attached to the skeleton, but not purely to the skeleton, as I'll show you in a minute or two. We will then go on in subsequent lectures early next week and the week thereafter to look at exactly how the muscle contract. We'll do that in to some extent excruciating detail. So let's begin with something simple. I will not look at the last two, I'll look at that next week, but I'll look at the first 5 or so on the top. So, the function, and I believe, I just saw the register, and I believe you had Dr. Shine in here already.
Yeah.
So some of this would, pretty much most of it I would think would be repetitious anyway. Okay, so, skeletal muscle, any muscle, as I was introducing a minute ago, has this ability to convert ATP, which is the chemical form of energy, if you want, into a mechanical movement. All right? And that mechanical movement I refer to as shortening. By doing so, you generate force. And if you generate force, you can pull on something or push on something, and therefore you can create movement. Again, muscles, if you look at my diagram, they're made up of these long cylindrical cells. The top picture is a photograph from a histology kind of view, if you want. And you have these long cylinders, if you look at it in terms of, like long tubes. And on the outside, on the periphery of the cell, is found the nucleus, so you can see the nucleus, and sometimes it's pushed to the corner, okay? You can also find mitochondria in these cells. But then if you look at this thing, if you look at it closely, it has a striped pattern. So if you're looking at the striped pattern, you've got like the dark pink and then the white, and then the dark pink and the white. You understand what I'm getting at? Okay? And that arrangement, if you want, is what is distinctive about skeletal muscle. And I'll tell you what that dark and light band or pink thing is in a minute or two. So again, skeletal muscle is associated with bones. It can also be associated with skin in a minute, with bones, with the skeleton, and so on and so forth. The ability of the muscle to contract or convert its ATP which it gets from the mitochondria into a mechanical force, which we now call contraction, is due to certain proteins it has in there. And you've come across this actin and myosin filament proteins, and I'll look at that in a little bit of detail in a minute or two. The arrangement of these proteins, or myofilaments, is what gives you this light and dark pattern. And if you now look at the diagram at the bottom, they took those two middle ones out and they put it in a diagram. So you've got the banding or striped patterns, or striated patterns, if you want to call it that. You've got a nucleus that's kind of pushed onto the outside with all of these things in the middle. And you will also have mitochondria there. Okay? So, let's take that one step further now. I mentioned that skeletal attached to bones, but you can also have attached to the face. And this skeletal muscle that I'm showing you here is attached to facial muscles, skeletal muscles on the face. They are essentially responsible for, you know, when you smile and when you move your mouth and so on and so forth. But in this particular case, I'm just showing you a problem, and the problem is called Bell's palsy. It's called facial nerve, which innervates these muscles. And in this case, it's called cranial nerve number 7. It becomes paralyzed. This happens when people get a viral infection and so on and so forth, and then, you know, it subsides after that. And in this particular case, the nerve is attaching, or the muscle is attached to skin. And obviously when that muscle will contract, the skin will move. But in this case, the muscle is not contracted, it's paralyzed, or for a short while, it's paralyzed. And it's called Bell's palsy. So if you're looking at this particular lady, she has this on the left-hand side. Notice when you ask her to smile, you as a doctor ask her to smile, that left-hand side is kind of not working properly. In other words, if you notice the eye, can you see the eye? Cannot close the eye, okay? The lips, she cannot pucker the lips. You know what does the word pucker the lips mean? Don't know what that word means? Huh? Yes? Yes. Yes, you guys use cosmetics to pucker your lips. People use Botox to pucker their lips. Okay? But she can't do that. All right? And obviously she doesn't smile on that left-hand side. So this is what is called Bell's palsy. Does it go away once the infection settles down? Yes, it does. Okay? So it's not only, the point I'm trying to make is it's not only, the skeletal muscle is not only attached to bones, you can have this kind of thing where you have facial expressions are impaired and so on. What does this word mean? You might as well get used to it now. What does the word ipsilateral mean? Huh? On the same side. In other words, the nerve on the left, for the lady on the left-hand side. Okay? Now, in neuroscience, this is a very important word, because sometimes when a nerve from this side of the body goes to this side of the brain, that's called contralateral, but if it goes to the same side, it's called ipsilateral. Okay, so the patient is unable to wrinkle their forehead, close their eyes, pucker their lips, and so on and so forth. And the muscle around the lips is called the orbicularis oris muscle, and she cannot contract that skeletal muscle. Okay? The same thing. The same thing applies to people who do Botox. Okay? on the side of their faces or on the forehead. It's called glabular lines or crow's feet. What you're essentially doing is you're preventing the muscle from contracting by injecting the Botox. And when I do neuromuscular junction, I'll tell you how that works. Are we okay so far? Yes. Good. So it's not just bones, it can also be skin. So although skeletal muscle, as the word suggests, is attached to bones and therefore brings about movement, there is also skeletal muscle, as you probably heard, during GI block for the upper third of the esophagus. It's skeletal muscle. You have skeletal muscle in the diaphragm. Okay? So it's not just purely two bones is what I'm getting at. All right? They cause eye movements. Okay? In other words, moving around, moving less, moving right, and so on and so forth. Again, movements of the body, and these movements, in the case of the skeleton, obviously, will allow you to walk. And you can have, I put a diagram here, and you can have flexion of the muscle. So if you are looking at the bicep, it's attached way up there on the shoulder, and we call that the origin of that muscle. Then it gets attached somewhere here on the ulna. That's the insertion. And when that muscle shortens, then the lower arm moves up. And we call that flexion of the elbow, or flexion of the arm at the elbow. Relaxation of the muscle will not allow the hand to move down. You need another muscle to actually move it down, and now you have the extensor, in other words, the tricep, which will now pull the arm down. So, again, you've got this movement that's taking place when the muscle contracts. Again, you know, it could be walking, running, jumping, whatever it is you guys do. For those of you who are going to become surgeons, for people who are going to become surgeons where you use your fingers a lot, okay? Again, the skeletal muscles are required in very, very delicate, fine movements. Now, the ones I've cited here, you know, writing, I do not know how many of you play the piano or the violin or the guitar for that matter. But again, for surgeons, it's a very essential feature to make those skeletal muscles work properly, okay? One last function of these skeletal muscles, apart from the movement at a joint, is because you're requiring ATP to bring about contraction, that means you can generate that ATP in the mitochondria. But you should know that the production of ATP also gives you a lot of heat, okay? Because if you take a molecule of glucose, it's only about 40 to 60% efficient in generating ATP. The rest of it is lost as heat. So when skeletal muscle contracts, it generates heat. So another function of the muscle is to maintain our body temperature, okay? So we are homeotherms, we need to be kept warm, and shivering and contraction of the muscle will generate that heat we require. Okay, so if you're looking at the muscle now, and I'll come back to the slide, I just want to look at the picture first. And again, I showed the bicep, okay, with its origin and its insertion on the ulna. That bicep is that entire muscle. It's a large, bulky muscle. And in the diagram, it shows you that little square kind of thing, or the rectangle thing, it's cut. And if you cut this muscle, you've got connective tissue on the outside, which I'm assuming Dr. Shahid called the epimysium, and connective tissue, by virtue of its name, will be, you know, things like dense connective tissue or loose connective tissue or whatever it is. you know, and so on and so forth. So that connective tissue which envelops the entire muscle is called the epimysium. And I put this little thing here to say, okay, that's the epimysium. If you cut open into that muscle, you will find bundles. You will find bundles, and one bundle is sticking out. And it's given a name, it's called a fascicle. Around that bundle is again connective tissue. Now we call that a perimysium. So dense, loose connective tissue, whatever it is found around that one bundle. It's not a muscle cell yet. Okay, it's not a muscle cell, it's a bundle. Around the bundles, you will find blood vessels, you will find nerves, and so on and so forth. So now you look at that one bundle, and you now pull out of the one bundle, you pull out one bundle, and now you have a muscle fiber. Are we okay so far? You have a muscle fiber. That muscle fiber now, within it, are found fine cylindrical kind of tubes. So let's look at the muscle fiber itself. It has a connective tissue around it. It's got its endomysium, and it's got its nucleus. That's because it's a cell. That's the basic muscle cell now. Not this, but that. So it's got its nucleus. It's got mitochondria. It's got its endoplasmic reticulum, and so on inside here. It has pushed its nucleus to the side because it's filled up with all of these little things. Okay? Now what are those little things inside the actual muscle fiber or muscle cell? They are called myofibrils, and there's one shown here. All right? And the myofibrils are made up of proteins or myofilaments. Myo, muscle, filaments, the proteins. And they are arranged in such a way that you end up with this banding pattern that you actually see in the muscle. So this is not the cell. This is a single cell, which is made up of, you know, I do not know how many you have in there, okay, myofibrils. And then that is now found inside a fascicle, and I do not know how many you have in there, and those bundles now will give you the entire muscle fiber. So let's go back now. So there we are. An entire muscle is composed of muscle cells called muscle fibers. Not the myofilament, the muscle fibers. Again, the length of the muscle fiber depends upon the length of the muscle, obviously from insertion to origin to insertion. Okay? They are multinucleate, these cells, the myofibers, they're multinucleate. In other words, they started off as single cells and then they fused to become like a syncytium. All right? Now, this is one of the few multinucleate cells we have in our body. That's muscle fibers. The other one is osteoclasts. Do you know what are osteoclasts? Huh? Yes, it's the cell that breaks down bone. Okay? Osteoclast, and the other one is the megakaryocyte, the one that gives you platelets. So there are just few of them that have, you know, more than one nuclei in them. So skeletal muscle is also abundant in mitochondria. Okay, so you've got this membrane around it, and you've got the nucleus pushed out to the side, but you also have lots and lots of mitochondria, which kind of makes sense because you require the ATP. Again, contraction requires a coupling between the biochemistry or the making of the ATP and the mechanical movement of the proteins. All right? And lastly, it involves a cyclical process of cross-bridge formation, which then gives you the contraction and then relaxation. So, anybody has anything to say about this particular thing? Are we okay? Yes? Good. So, let's now look at this thing with its mitochondria and so on. So there's one muscle fiber, okay? And there's these cylindrical things that are sticking out of it. It's almost like, you guys like that wafer thing with the chocolate filled in it? Yes? You know what I'm talking about? Everybody seems to make it these days. You have a Dove version or I do not know, a Galaxy version and a whatever kind of version of it, okay? And that's what you're looking at. Now, if you're looking at this thing, you got this endoplasmic reticulum kind of thing, which is represented in blue. It's given a special name because it's in the muscle. It's called the sarcoplasmic reticulum. So I put the fancy words on the right-hand side. So the membrane on the outside, the gray, is the sarcolemma, which is plasma lemma, okay? Then, you would notice that the plasma lemma actually goes into the muscle. What do I mean by goes into the muscle? It goes in like a tube like this, it goes like this, like this, like this, and then it comes out the other side. In other words, the fluid that's inside the T-tubule is actually extracellular fluid. Okay? So there's extracellular fluid here. Whereas the fluid here would be intracellular fluid. In other words, you have a high concentration of sodium in this fluid. You have a high concentration of potassium in the intracellular fluid. So just keep that at the back of your mind. So okay, we got the T-tubules going in. Notice it's going around, you know, the galaxy tube, or the chocolate-filled galaxy tube if you want. And the other thing is you'd find mitochondria. There's mitochondria, and obviously the nucleus is pushed out onto the outside. I can't see it in this one. Okay? All right? Now, also notice that there is a kind of a special relationship between the sarcoplasmic reticulum and the T-tubule. They kind of meet each other here like this. And this rounded version of the sarcoplasmic reticulum is called the cisterna. There's the T-tubule, and then you've got another cisterna on this side. And that's also the sarcoplasmic reticulum there. Okay? So I put a blurb. So sarcoplasmic reticulum, sarcoplasm would be the cytoplasm. Sarcoplasmic reticulum is like the endoplasmic reticulum, but it just does one major function. It stores calcium. The T-tubule would be the invagination, if you want, of the sarcolemma. The myofibrils, those are these little things, would be the actin and the myosin filaments, which I'll come to now. Okay? So, there's that same kind of picture from another textbook on the top. Again, you've got the sarcolemma, you've got the hole that's going in. There's the T-tubule coming in on the top, and then it's going, the yellow thing that goes around, go behind that one, in front of this one, and then it comes in here. And this is the cisterna of the sarcoplasmic reticulum. And this arrangement of cisterna, and T-tubule is called a triad. Okay? Now these names, I'm assuming Dr. Shire has already kind of gone through with you. Okay, so now if you look at the chocolate-filled wafer, there we are, and it's made up of these myofilaments. Myofibril, myofilament. The protein is a myofilament, and if you look at the myofilament now, these ones that are attached to the Z-looking kind of structure, or the Z line, is actin. There's actin. There's Z line, and that's the actin. It's a double helix. It's a double helix protein that's attached to the Z line, and it extends into that one into that cell, this one into this cell. Okay? So you have actin, and the one in the middle, the dark one, the grayish blue one, is called myosin. And this is myosin. which is right in the middle of the sarcomere. That's the structure it makes. So there's myosin. On the edges of myosin, or the ends of myosin, you have these projections sticking out, and they are called cross-bridge heads of the myosin. And I've given you a diagram down here. So you have this tail region of the myosin, a little hinge kind of thing, and then the head of the myosin. Whereas actin is just made up of these round, ball-looking like structures there. Let's just look at the actin and the myosin in a little bit more detail. So we'll do myosin first. Again, it has a tail. There's the tail part. Again, it's a protein. Remember, it's a protein. So it has a tail. And then you've got a hinge kind of region called an S2 region. Don't worry about S2 and S1. And then you've got a head. And this head region has two important functions. Number one, it has a place where it will attach to actin. And number two, it has a place where an ATP can bind to it. In other words, there's an enzyme here. And the purpose of that enzyme is to hydrolyze the ATP. So I put a couple of things for you. Myosin, again, in that region of the sarcomere where you would find it, it's called the A band or the dark band. All right? The myosin molecule is made up of this tail and the head. The head region has the actin and the ATP binding site. When calcium is present, the myosin head will attach to the actin. When you take the calcium away, it will detach from the actin. On the other hand, you have an ATP binding site. So ATP, when ATP binds to the myosin, then the head will detach from the actin. Okay, so calcium allows the binding of the head to bind to the actin. ATP binding allows detachment of the head from the actin. When you hydrolyze the ATP using the myosin ATPase, that does two things. Number one, it will move the head. So if this is actin, it's attached, the myosin head is attached to it. My right hand, okay, or fist is the myosin head, it attaches, okay? And then what it does is it pulls. When the ATP attaches to it, it detaches. When ATP hydrolyzes onto it, it then goes back to this position. And that back to this position, like the 45-degree angle, is what we call the cocked position, where it's ready to contract again. Okay? So that's our friend myosin. What about our friend actin? Again, it's made up of a globular protein, these round little blobby things, and they are arranged in a helix. So there's filamental, F stands for filamental actin. It's almost like the maspa, you know, the beads, that kind of thing. All right? And then it's held together by a the black line, there's the black line up there on the top, called tropomyosin. And attached to tropomyosin at intervals is a group of proteins called troponin. Troponin T, troponin I, and troponin C. Okay, and you can see them here. So there's the actin, the myosin, tropomyosin is the black line, and this is the group of troponins. 3 of them. Troponin, C, T, and I. What does C do? C will attach to calcium. Okay, what does T do? T attaches to troponin. Tropomyosin, sorry. What does I do? It inhibits the attachment of the binding of the actin to the myosin site. Okay, and that's where the I and the T and so on comes down. And because of this intimate relationship between troponin and tropomyosin, it actually regulates the cross-bridge formation, which is next week's lecture. Now, because you're becoming doctors, we need to complicate your life a little bit more. There are some other proteins here. So if you're looking at one sarcomere, there's a Z line and there's a Z line. All right? Attached to the Z line is actin, there's another actin, there's another actin. Who attaches the actin to the Z line is a protein called actinin. It's written there. Actinin attaches actin to the Z line. Okay? Who keeps this thing straight? Keeps the actin straight is this protein here, okay, called nebulin. One more protein, we got the myosin filaments in the middle. There's the myosin filament. Okay, 1, 2. Who keeps the myosin filament straight in the middle? That's our friend titin. Can you see titin? Okay, titin is a large elastic protein that makes sure that the myosin is in the center all the time. Okay, so you've got these proteins that are kind of regulating this thing within the sarcomere. Now, the one I want to emphasize is this one, and that's this protein called dystrophin. Now, understand something here. If this is a muscle cell, and these things are becoming shorter, longer, in other words, contracting, relaxing, that force must be supplied or applied to the connective tissue on the outside. There's nothing on the outside that's contracting and relaxing. This thing is contracting and relaxing. Are we okay? So we need to transfer this force to the outside. That collagen and the fibroblast and the dense connective tissue and so on. We need to transfer it to that. Who does that? Dystrophin. Dystrophin together with this huge complex will transfer that force to the outside and pull this thing when the muscle contracts. What happens if you don't have it? And now you have muscular dystrophy. Okay, so muscular dystrophy, again, mostly in men because it is sex-linked. It's on the X chromosome, and since men only have one of them, and if they have a defective version of it, they will have muscular dystrophy. Again, affects males early in childhood. Generally, they will, when they are born, they're okay. And early infancy, the muscles of the lower limbs will start to become weaker and weaker, and then it works its way up. By about the age of about, 20, 24, 28, it affects the respiratory muscles and then they die. Okay, dystrophin is a protein that will supply or transfer the force from the muscle cell to the outside, and patients with Duchenne, that's the guy who discovered it, Duchenne's muscular dystrophy, have little or no dystrophin, and therefore the muscle becomes weaker, and obviously these patients die in early adulthood. Let's now look at those myofilaments, the actin and the myosin. So if you're looking at a diagram, there's a Z line, there's a Z line. Attached to the Z line are the actin, and in the middle you will find the myosin. Okay? So according to my notes, the muscle fiber is divided into several of these, what we would call sarcomeres, thousands of them, okay, in one myofilament. And you have hundreds of myofilaments inside a muscle fiber. So, again, each myofibril is an alternating light and dark pattern. At the center of the dark band is the A, or the myosin molecule, the A band, and in this case there's the A, there's the A up there, okay? And between one A and the next A you have the I, there's the I. In the middle of the I you have the Z line, where the actin is attached to it. In the middle of the A, you have the H region, where you do not have any overlap between actin and myosin. And then in the middle of the H, you have this thing called the M line. So there's your myosin, okay, the dark band. There's your light band or I band. There's I here and I here, okay. This would be your H zone, your M line, and that's your Z band. Okay, that's how that arrangement kind of works. So the I lies between the A bands, the Z line separates the I into two, okay? Obviously, one to one sarcomere and the other one to the other sarcomere. The repeating unit of the myofibril is called a sarcomere, and you have hundreds of them in a myofilament. So if you look at that in terms of a diagram now, there's it here. So you've got Z line, attached to it is actin. In the middle of it, the A band is myosin. In the middle of the myosin, you have the H zone. Okay, the H zone where the actin and the myosin are not overlapping. In other words, from there to there kind of thing. Now, if you look at it in a cross section, if you're looking at the overlapping region, you notice that for every one myosin, there are 5 actins around it. Did I get the number right? 6. Yes? 6 actins around it. Okay? If you are looking at only the actin, obviously there's no myosins there. If you are looking, there's the actin and there's the myosin on the other side, the A band and the I band. Okay? And I want you to keep this thing in your mind, because when I come to it later on, I'll tell you why I emphasize this particular thing. Okay, so I wrote the same thing down for you. Again, actin attaches to the Z disc. I band represents where you have the actin only. A band is the entire length of the myosin. At the edges of the A band is where you have the overlap or the cross bridges. The H zone is in the central region of the A band, and the M line is a protein band slam dunk in the center of the myosin molecule. There's an electron micrograph now. So again, M line, there's it in the middle here. Okay, H zone, that's the H zone, all right? Here you're getting the overlap between actin and myosin. So this whole thing will be the A band, and this would be the I. Here's the Z line, and it's shown to you at the bottom. If you increase the overlap between actin and myosin, the overlap between the myofilaments, then the sarcomere will become shorter. Let's look at what happens when you do that. So there we are. We do not have too much of overlap between actin and myosin, or we do not have too many cross bridges between actin and myosin. What do I mean by cross bridges? There's myosin, there's a cross bridge, it's attached to actin. And then it will pull the actin. This side will pull on this side. Okay, so it will pull onto the actin. So there's your overlap. Notice the first diagram, there's very little overlap. The sarcomeres are rather wide, therefore the length of that myofibril is longer. Now you're getting more overlap, there's the greater overlap. Notice the myofibril is becoming shorter. Now you have even more overlap, and now the whole thing is even more shorter. Why? Because this is constantly pulling on the actin, okay? And it is this overlap and pulling on the actin that pulls on the Z line and therefore shortens the sarcomere. But because the sarcomeres are in series, and if all of them are shortening together, you have a multiplying effect, and therefore you will have a rather large shortening of the entire muscle itself. Okay. How that happens is obviously next week. Let's just quickly look at the sarcoplasmic reticulum very quickly. Again, there's a muscle cell, that's extracellular fluid. There's a T-tubule now, okay, which has extracellular fluid. In other words, high sodium, low potassium. This is intracellular, low sodium, high potassium, okay? There's a sarcoplasmic reticulum, there's a T-tubule. So you're looking at this contact between the terminal cisterna of the sarcoplasmic reticulum and the T-tubule. So there's it blown up here. And you notice there's the T-tubule. You have a protein. Okay, you have a protein in the sarcoplasmic reticulum that's going to kind of release calcium. So there's the T-tubule. There is this protein, it's called a DHP, or dihydropyridine receptor, that will tug onto or pull another protein that's called a ryanodine channel. And when that opens, the calcium will come out. But that detail is for next week. Just showing you the structure. Again, once the calcium comes out and brings about contraction, it needs to be quickly pumped back in. So on the membrane of the sarcoplasmic reticulum, you have a ATPase, it's called a SERCA, sarcoendoplasmic reticulum. Okay, ATPase, it pumps the calcium back into this thing and thereby keeps the intracellular calcium low. All right, and that's when the muscle will relax. So again, sarcoplasmic reticulum regulates intracellular calcium. T-tubules will pass close to it. You have the SR is the intracellular network, the T-tubule is the extracellular network, okay? And then the part of the sarcoplasmic reticulum closest to the T-tubule will be called the cisterna. So this would be a cisterna. There'll be another one on this side, and all three of them together is called a triad. Now, to make a muscle contract, you need an impulse. You need an action potential. Yes? Where do you get that action potential from? Because the muscles we will talk about are not involuntary, they are voluntary. Okay? So, the impulse comes from the spinal cord. It comes through a motor neuron to a muscle. So there's the muscle you're looking at. You know, your fascicles, and now you've got your muscle fiber, your individual fiber. The motor neuron will come in, will then branch off, and each muscle fiber will get one branch or one process of that axon or that motor neuron. Each, so if you're taking this one, there's the one that's shown here. So each one of them has one of these branches of the motor neuron axon. Okay? So the motor neuron can come in and branch into 2 or 3. That branching is called the axon process. It will come and end up sitting onto a muscle fiber. The muscle fibers that a motor neuron innervates or goes to or excites, that neuron plus the fiber is called a motor unit. Are we okay? A motor unit will be the motor neuron plus the fibers that it innervates. We have small motor units, in other words, an axon with a few muscle fibers. Generally, small motor units are used for fine movements, like a surgeon. Large motor units, where there are lots of muscle cells activated, are used for things like jumping and running and so on and so forth. Which brings me to my last slide. We can have motor units. The top one is showing you one motor unit because I've got one motor neuron going through to all those fibers. Whereas the bottom one is showing you 2 motor units. Obviously, the pink one is going to the pink fibers, the orange motor neuron is going to the orange fibers. Yes? Are we okay? And we can have small motor units where the motor neuron can go to fewer fibers, or large motor units where the neuron goes to lots of fibers. Okay. I will see you next week. Have a good weekend.